explain the differences between blood, tissue fluid and lymph. This vessel here is a capillary. It's a very thin vessel with a very small lumen and very thin walls that are 1 cell thick. And the vessel contains a range of different cells, including erythrocytes or red blood cells and leukocytes, which are white blood cells, and these include macrophages. Now the blood also contains platelets, which are cell fragments, and these help in the clotting process when someone is cut. Now the blood also contains proteins, and these include plasma proteins, which are called complement proteins that help in the immune system, and also hormones, which are responsible for signalling around the body by binding to receptor sites. Now you'd also expect in the blood for there to be a large amount of the nutrients that bodies need. And these dissolved substances are going to be too small to show on a diagram, but they include fats transported as lipoproteins, glucose in relatively small amounts, unless someone is diabetic, and amino acids formed from the body digesting proteins. Now in most of your body, the bloodstream would also include lots of oxygen obtained from the alveoli in the lungs, and because of the continuous maintenance of a steep concentration gradient due to the flow of blood, there won't be very much carbon dioxide in the blood, and that is the composition of blood. Now let's think about comparing the components of the blood with the components of tissue fluid. Now tissue fluid is a fluid that bathes the tissue in your body, and it allows the transference of nutrients and oxygen from the blood to the tissue, as well as the movement of waste products from the tissue back to the blood. So let's go through step by step. Will the tissue fluid contain erythrocytes? Well, the red blood cells or erythrocytes are very large. They cannot fit through the capillary walls, and therefore the tissue fluid should not contain any erythrocytes. Now, of course, if you have a small capillary burst and the erythrocytes can leak out, that can cause bruising. But if a large number of erythrocytes leak out, that could signify internal bleeding, which is a bad thing, obviously. However, these leukocytes, or mainly macrophages, you can see they've got a much more flexible shape, and they're actually able to squeeze through gaps in the wall of the capillary and enter the tissue fluid. And this allows them to fight bacteria or viruses that may have infected your body and are damaging your tissues. So macrophages can leave the capillaries into the tissue fluid, so you will find macrophages only, but not as many. Now in terms of proteins, you will still find some proteins in the tissue fluid, mainly either proteins that are secreted from the tissue cells or hormones that leave the capillaries and then bind to receptor sites on the target cells. Now that's particularly important in the liver and muscle cells, because obviously insulin has to act on those cells, which then causes them to absorb a large amount of glucose. Now large fats cannot leave the capillaries, and therefore you won't find fats in the tissue fluid, and things like LDL and HDL, or low and high density lipoproteins, will not be present in the tissue fluid. Now the other nutrients, glucose and amino acids, there'll be a small amount in the tissue fluid because they will be constantly diffusing from the blood to the tissue fluid, but these cells here will be rapidly absorbing the nutrients, and therefore there'll be much less amino acid and glucose in the tissue fluid compared to the blood. And likewise, there should be far less oxygen inside the tissue fluid compared to the blood, because the cells, whilst respiring, will be absorbing the oxygen via diffusion and using it to respire for energy. But during the process of respiration, these cells will be producing water and carbon dioxide, and this means that the amount of carbon dioxide in the tissue fluid will increase. Now, some tissue fluid will drain back into the capillaries, but some of the tissue fluid will drain into another type of vessel called a lymphatic vessel. Now, a lymphatic vessel is part of your lymph system, and the tissue fluid will drain from here, from the tissue, into the lymphatic vessel. And the idea is that the lymphatic vessel will actually act as kind of like a alarm system. So when the lymph flows through the vessels, any pathogens that are present are picked up by white blood cells, which can then mount a defense. Now, lymphatic vessels contain one type of cell, which are lymphocytes, which may include B cells, and also the lymphatic vessel will contain some of the proteins that drain from the tissue fluid into the lymphatic vessel. So some of those hormones, for example, will drain from tissue fluid into the lymph system. And there will actually be a high amount of fats inside the lymphatic system compared to the blood, because the body actually absorbs fats from the intestines into the lymph. And like tissue fluid, there will also be a smaller amount of oxygen, a smaller amount of glucose, and a smaller number of amino acids in the lymphatic vessels, because of course the tissue has already absorbed those nutrients and oxygen before the tissue fluid manages to get inside the lymphatic vessel. And of course there will be equally large amounts or larger amounts of carbon dioxide in the lymphatic vessel and the tissue fluid compared with the blood, because these cells here, these tissue cells, will have produced carbon dioxide, and as the tissue fluid drains away, it carries that carbon dioxide with it. So now we can compare the constituents of tissue fluid, lymph and blood. We're now going to describe how tissue fluid is formed from the plasma in the blood. So let's consider the water potential in the blood compared to the tissue fluid. Now, as we discussed, there are far more substances suspended or dissolved inside the blood compared to tissue fluid. For example, there are more blood cells, white and red, and there are more nutrients inside the blood compared to tissue fluid, and there's also going to be a greater number of different proteins inside the blood compared with the tissue fluid. And that means that there is a lower water potential inside the blood capillary compared with the tissue fluid, which would have a higher water potential as it has many fewer substances dissolved or suspended inside it. So the overall effect would be that the water would move via osmosis from an area of high water potential to an area of low water potential. And these arrows represent that direction of travel, from tissue fluid to blood. However, there's another major force or pressure that we have to consider before we can get an idea of how tissue fluid is formed, and that is the idea of hydrostatic pressure. So as the heart beats and pumps, it pumps blood through your arteries and obviously through the arterioles, which connect to your capillaries, and that's at high pressure. And that force of water, or plasma really, pushing the sides of the blood vessels creates hydrostatic pressure. So the capillary end nearest the arteriole will have a high hydrostatic pressure in the direction from the blood to the tissue fluid, because the blood is being pushed towards the walls of the capillary. And I'll draw that hydrostatic pressure arrow in green. Whereas at the venular end, where there is much lower pressure because the blood is returning back to the heart and the venules and veins have a much wider lumen, there will be a much smaller hydrostatic pressure, as shown in the green arrow here. So at the arteriole end, the huge hydrostatic pressure is much larger than the effective water potential, so overall fluid is pushed out of the capillary and into the space between the capillary and the tissue, which is called tissue fluid, and it carries with it dissolved substances such as amino acids and glucose to be absorbed by the tissues. Whereas at the venule end, under much lower hydrostatic pressure, there is an overall influence of the water potential, which is larger than the hydrostatic pressure, so tissue fluid then is reabsorbed by the capillary, and likewise dissolved carbon dioxide is carried across from the tissue fluid to the capillary, and this will then be pumped back to the heart and then to the lungs to be expired. So in summary, at the arterial end of the capillary, there is a high hydrostatic pressure due to the pumping action of the heart. This hydrostatic pressure is larger than the effect of water potential, and therefore fluid leaves the capillaries to form tissue fluid. Whereas at the venule end of the capillary, there is a lower hydrostatic pressure because the vessels are further from the heart and are also the venules and veins have a much larger lumen. This means that there's a smaller effect of hydrostatic pressure compared with the effect of water potential, and therefore the water moves from the tissue fluid back into the capillary, carrying carbon dioxide with it. So we know that tissue fluid has a higher water potential than the blood. Can you explain why? Now the reason is because the capillaries have small gaps in the wall that allow only some substances to leave, and the larger substances like proteins and blood cells cannot leave the capillary to go to the tissue fluid. And that means the water potential of the tissue fluid is less negative than the water potential of the blood, and therefore the water potential tends to push fluid from the tissue fluid back into the blood. And this will carry across the carbon dioxide produced during respiration.